Our penultimate session about to kick off. This one is going to focus on microglial medicine, so some potential therapeutic approaches to modulate microglia and neuroinflammation in different disease states. My name's Hugh Nuttall. I work at Eli Lilly and Company, a US-based pharmaceutical company. I'm in their neuroscience team, but I'm part of the kind of business development and licensing group. So always looking for external opportunities to work with groups and companies in this area. So if any of you have some great ideas you'd like to look for a partnership with us, please do get in touch. It's a great pleasure actually to introduce my colleague Yaming Wang, who's going to kick us off for this session. Yaming is based in Indianapolis, our headquarters. Some of you may know him from his previous work prior to joining the company, where he's part of Marco Colonna's team at WashU and published a number of papers on TREM2 function. So, over to you, Yaming.
All right, thank you, Hugh. And first, I want to say thank you to the organizers for this opportunity to share our work with the broader community. It's been a fantastic meeting. I think from our panel discussion yesterday, right? So I hope we all acknowledge that it's really important for us to understand the pathophysiological functions of this microglia subsets, right? Mostly identified from the transcriptome analysis. I think this is particularly important from a drug discovery perspective. We need to understand how do those microglia contribute or influence the disease pathogenesis. So today, I would like to share some of our work aiming to decipher how microglia influence the Alzheimer's disease pathologies. All right, so this is my conflict interest statement. And so I don't need to introduce too much on Alzheimer's disease, right? So we know that it's the most common form of dementia, and is characterized by the extracellular deposition of amyloid plaques, as well as the intracellular deposition of the tau tangles. But importantly, we know that the deposition of these two pathologies happens in a sequential manner, mostly from the PET imaging, longitudinal PET imaging data in the living patients. We know that amyloid deposition is necessary for the subsequent development of tau pathology, and neurodegeneration follows. We know that the Alzheimer's disease has multiple influencing factors. Particularly, aging is the most important factors for dementia as well as Alzheimer's disease, right? And also, as Angela talked about genetics, right? Alzheimer's disease is heavily influenced by human genetics. And in addition, I think it's really nice for the field in the past few years that there's a breakthrough in disease-modifying drugs in slowing the disease progression. But we know that those patients, even after the treatment, they still continue to decline, right? So there's still lots of work to do. Don't need to introduce this slide. So I think we're all familiar, and I don't think there's any argument that we believe that microglia plays a critical role involving in the pathogenesis of Alzheimer's disease, right? So this is the pathway for TREM2, right? So I think not only TREM2, but also the modulator of a TREM2 signaling, as well as the signaling components, all have been implicated in the pathogenesis of Alzheimer's disease as a risk factor. So I think one of the critical interest for myself, as well as for in both from a biology perspective as well as from a drug discovery perspective, is really understand how does TREM2 affect the microglia functions in disease, right? So before I want to start with a little bit of history. So this is my interest in TREM2, probably all the way dated around 2012, about the time I was finishing my PhD in Marco Colonna's lab. So at that time, the lab has been working on TREM2 about 10 years since they first cloned in the lab, right? So and unfortunately, at that time, we didn't really know a lot about the biology of TREM2. We know it's important because TREM2 deficiency leads to nasopharyngeal disease, right? But really, in mice, at least in the naive TREM2 deficient mice, there's really nothing. I mean, the phenotype is really, really subtle, right? So, it wasn't very clear how, what's really the function of TREM2. But I think everything changed. So there's one day, Alison Gold, who was also at WashU at the time, our upstairs neighbor, actually organized a meeting and shared some really exciting data that eventually became the seminal New England Journal of Medicine paper that identified TREM2 as a major risk factors for Alzheimer's disease. So we thought maybe this is really the model or the system that we can use to evaluate the functions of TREM2. So essentially, naturally, actually, we crossed the TREM2 deficient animal to 5X FAD mice. And then one of the really striking differences almost immediately when we start analyzing the mice is that, unlike the wild type 5X FAD animals where the microglia is heavily associated with amyloid deposition, in the absence of TREM2, those microglia, no longer seems to be interested in the amyloid pathology, right? So you can see it's very obvious from this is actually probably the first confocal studies that I did in those animals. We also evaluated the transcriptome analysis. So this is the time, single-cell RNA-seq was still at infancy. We actually did the microarray studies using purified microglia, because that was the most established method at the time, right? So what we can see is that you do see this cluster of genes that are upregulated in the 5X FAD mice, microglia, right? And also another cluster of genes that are downregulated in the 5X FAD microglia, right? And the interesting thing here is that in the absence of TREM2, that this transcriptome switch was no longer observed. So of course, through the work with Ido Ahmed as well as Oleg Butowski, we know that those are the signatures now correspond to disease-associated microglia, or IMGND, as well as the homeostatic microglia. Right, so that was very interesting. So this data really tells us that TREM2 is really the trigger that modulates the switch of these two phenotypes. So the next question we always want to ask is, what's really the function? Why microglia? What's the function of microglia doing around the plaques, right? So one of the really striking observation we made at that time is that the plaque morphology is very different in the absence of TREM2, at least in this 5X FAD mice. They're much loosely compacted and also seems to, very sort of diffuse looking, if you will. Right, and further collaboration with Jaime Guzendler's group at Yale, we further confirmed that a critical role of microglia in terms of compacting the plaque into a more a compacted form as a way to potentially restrict amyloid toxicity, right? So that was our work, when I was at WashU. So when I started my group at Eli Lilly, this is a continual one of the areas I was interested, at least from a science perspective, right? So one of the lingering questions is that does TREM2 mediate phagocytosis of A beta? I think this is one of the key questions. And then the in vivo data from different animal models as well as different time points, it seems to be inconsistent, right? So in some models you see increase, some other models you see decrease, and also depends on the time points. So we also want to, at this time, I want to evaluate how does TREM2 affect the Alzheimer's disease, A beta pathology in the mouse that as close to sort of this sporadic A beta deposition as possible, right? Because of, the 5X FAD mice have 3 mutations in the APP genes and 2 mutations in the PS1 genes. It's a very artificial model. So I want to have a model that seems as physiological as possible. So with this case, we picked the J20 mice, which only has a Swedish mutation. So the downside of this is that it takes a long time for the deposition to occur. Right, so here we actually have to wait to at least 12 months for the initial amyloid pathology to occur, and then it's not until 18 months you start to see appreciable amount of A beta in the brain. And then when you look at the A beta load, so this is utilizing an antibody specifically targeting the A beta peptide. We found that actually TREM2 did not really affect the total A beta load, right? This is by both histology as well as biochemistry. However, if you look at amyloid, this is using methoxy X34 or thiol-T, so this is really to detect the aggregated beta sheets, right? We found that actually we see an age-dependent reduction in X34 intensity over time in the TREM2 deficient animal. I think this is further consistent with the initial observation in the 5X FAD mice that the TREM2 mediated microglia function is really important in terms of compacting the plaque into a more concentrated form. Another interesting observation is that when we look at the formation of dystrophic neurites, so those are the sort of this abnormal axonal varicosity around the amyloid plaque, presumably is the consequence of amyloid toxicity. We found that there's a significant amount of increase in neuritic dystrophy in the TREM2 deficient J20 mice. I think further indicating that the consequence of this compaction, a protective consequence of this compaction, right? So in the absence of this TREM2 mediated microglia response, you further increase the amyloid mediated toxicity, right? So this is good. So then what's next? How do we connect the dots, right? So we have amyloid, and can we demonstrate this has downstream consequences in terms of facilitating the subsequent spread of tau pathology? Right. So to understand this question, we utilized a model initially developed by Virginia Lee. Right. We injected purified seeds from 80 brain samples into the J20 mice. So if you inject the 80 tau seeds, say in the hippocampus, so immediately the seeds are able to seed into the neurites. Mostly in the dystrophic neurites around the plaque. This is the place where you see a lot of ATA signal, which is antibody against hyperphosphorylated tau protein. Right. So if you look at the TREM2 knockout mice, the J20 mice, you can see that you can appreciate that you do indeed see a lot of increase in seeding, right, in the hippocampus where the 80 tau seeds is injected. Another interesting aspect about this model is if you wait long enough, that this initial seeding aggregation induced from the seeding activity can also spread into anatomically connected regions, such as the hippocampus in the contralateral side, entorhinal cortex, as well as the supramammary nuclei. Right, so if you wait long enough, and you can see how does TREM2 deficiency affect the spread of tau. And then I think it's also nice to see here is that not only this increase in dystrophic neurites in the TREM2 deficient animal exacerbate the initial seeding of the tau, but also facilitate the subsequent spread of tau pathology. Right, so I think this is really argues that this TREM2 genetic risk factor is really controlling the amyloid toxicity, which eventually facilitate or controls the subsequent spread of tau pathology into other regions. Right, so this is good, right? I think this is particularly you can put where this TREM2 genetics onto the amyloid cascade, right? However, when we're considering translation from genetics targets to therapies, I think there's just more we need to think about, right? Particularly, if you want to treat AD patients that are symptomatic. So most likely those patients, they're old, right? So there's ongoing aging. And not only they have a full brains of amyloid, but they also have a lot of tau tangles, and as well as ongoing degeneration. So if you put a therapy into those patients, I think you better understand how does the therapy impact other pathologies in the brain, right? So this is where we'll spend quite a bit of time to understand what's really the impact of TREM2 modulation during aging, and how does it modulate the tau pathology, because once the tau spread into a region, that there's also a local accumulation, local spread of tau pathology, right? So what's really the function of TREM2 modulation or the microglia response on the local tau spread, as well as tau-mediated neurodegeneration, right? So there's different aspects of things that we need to consider. So what do we know about this DAM or disease-associated microglia on tau-mediated neurodegeneration? So this is a review from Dave Holzman. We know that at least based on literature, there's a few potential possibilities, right? So there's has been data demonstrating that this DAM microglia potentially promotes the local tau spread, likely through the spread of tau in the exosomes. In addition, DAMs can potentially induce neurotoxic astrocytes, as well as phagocytosing neurons. I think Guy has shown some very interesting data from that aspect, right? So to really understand this in the context of tau-mediated neurodegeneration, particularly from a therapeutic perspective, so we actually develop a TREM2 antagonist antibody, right? Really try to understand in response to therapy, How does the microglia response change, and how does it influence the pathologies in the disease? So to do this, we actually utilize RTG4510 tau transgenic animals. So those are the expression of FTD-P301 L tau, which leads to the development of tau pathology as well as the neurodegeneration. What we found is that if we do a single dose injection of the TREM2 antagonist antibody into the TG4510 mice, we know that this mice at 7 months of age is already have loss of tau pathology, have ongoing neurodegeneration, and this antibody treatment can really, even a single injection can significantly reduce amount of the DAM microglia marked by the expression of CLAC7A, right, which is one of the key DAM genes. And also interestingly, we see actually an increase of a P2Y12 positive microglia. So this is a microglia associated with homeostatic signature. Right, so I think this is really tells us that maybe we can actually suppress the formation of the DAM microglia in the mice. We also confirmed this by gene expression analysis, and you can see here that we do see a very nice suppression of genes associated with DAM, as well as a significant increase of genes associated with microglia homeostasis. Right, so this is really good. And also, if you treat the antibody chronically in those mice, you essentially can reverse all the DAM microglia back to homeostatic state, right? So this is good. Then I think we have a system. Then we can really understand what's the consequence, right? If you suppress the DAM microglia in the context of tauopathy. So this is really depends on how when do you treat. So if you treat the mice at the onset of tau pathology, right, very very early, around 10 weeks of age, we actually saw a significant reduction in tau pathology. This is after 8 weeks of treatment, right? However, if you treat later, when the tau pathology is already established and neurodegeneration start to occur, for example, if you start to treat at 16 weeks of age, so we actually no longer saw any impact on tau pathology, but rather we actually see, it seems that the antibody treatment can rescue the neurodegeneration marked by the brain atrophy. And in addition, we did a little bit of functional analysis, particularly looking at the hyperactivity of those mice. This is a very prominent in these animal models. So if you do this in a running wheel, I think on average, the mice actually run about 1.6 miles a day. I think it's equivalent to 2.5 kilometers. Certainly, longer than what I run every day, right? So if you do an open field, right? So we can really see is that at the beginning of within this, if you within this 16 weeks of period, if you treat the mice with isotype control, you start to see a significant increase in hyperactivity, right? But however, when you treat the mice with a 3CO with a TREM2 antagonist, this increase in hyperactivity was no longer observed. I think this also tells us that inhibition of the DAM microglia could also impact the function of these mice, likely associated with neurodegeneration. I think this is all good, right? This is, now we think maybe this is a therapy we can treat tau-mediated neurodegenerative type of conditions. I think this always happens, right? There's one actual experiment you do that changes your mind. So, as a pharma, right, we're interested in translational biomarkers. So at that time, neurofilament light chains, has been a very promising biomarker indicative of neuronal injury, right? So we know that neurofilament light chain is a structural protein highly expressed in the axons, in the axons of neurons. Upon injury, the neurofilament light chain NFL is released and then can be detected in either CSF or blood. And then we know that in numerous neurodegenerative conditions, NFL is upregulated. And also in response to therapy, such as in the case of MSA or MS, NFL is also a good biomarker for therapeutic efficacy. Right? So we thought, it'd be nice to demonstrate the NFL reduction after this antibody treatment, right? However, to our surprise, instead of a reduction, we actually saw a significant increase after the antibody treatment. Right? So that was very interesting. And also we did another study where we treated mice subchronically, where actually we didn't, this increase in NFL was no longer observed. So this suggests that somehow this chronic inhibition of the disease, the DAM microglia, could have a consequence in terms of, modulating neuronal injury. So given that NFL is an axonal marker, right? So we think that it might have something to do with modulating axonal health. So to do this, to look at this one step further, we went back to the TREM2 knockout mice, right? We actually did a longitudinal study, just looking at what's the NFL change over time. We know that NFL is a biomarker that elevated during aging, right? And then in human, and also this is very nicely to see that this is the case as well in mice. But also very interesting to see that the TREM2 deficient mice have a further elevation of NFL during aging, this aging process, right? And then the magnitude is very similar to what we have seen in the TG4510 mice after the antibody treatment. So this suggests that likely this disease-associated microglia somehow can limit this age-dependent axonal injury that happening spontaneous, right? And then we further confirm the consequence of this NFL elevation by performing a silver stain. So this is a special form of silver stain called Diomo stain that detects degenerating axons. What we found is that in the white matter of those mice, particularly in cerebellum over time, if you don't have TREM2, we detect a significant amount of the silver positive deposits that reminiscent of those kind of degenerating axons, right? So if you look at the image in the insect, you can see this kind of axonal varicosities, the axonal spheroids formation are really indicative of degenerating axons. And so if you quantify this, we were able to detect a significant increase in axonal degeneration at 2 years old period. And it's also very interesting that despite we see an NFL increase at 12 and 18 months, we don't see any pathological differences. I think this is really suggesting that this initial axonal injury that induced by some kind of aging process can lead to subsequent axonal degeneration. And the DAM microglia that induced by TREM2 is particularly important in limiting this kind of age-dependent axonal injury, right? So when you put everything together, it is actually, we think that there's actually different aspects of a DAM microglia during the disease pathogenesis, right? So we know that likely this DAM microglia is critical for healthy aging, right? So if you don't have, one is that in the white matter, this microglia limits age-induced axonal damage, right? So, in the case of not so high color disease, if you don't have TREM2 at all, you do see a very severe axonal white matter degeneration type of pathology, right? So this is if you don't have TREM2 at all. But if you have a little bit reduction in TREM2 signaling, maybe for the case of R47H mutation, I think this is where you might have a more vulnerable to amyloid pathology, right? Particularly, the amyloid likely to be more toxic, and this could further facilitate the subsequent spread of tau into other regions. We know that this is really a key hallmark of the onset of the disease. However, once the disease is onset, right? So you have all this degenerating process ongoing. This DAM microglia might not be so good, right? So it could contribute to the local tau spread, as well as some downstream of tau-mediated neurodegeneration, although we still don't really know the exact functions of those DAM microglia in mediating this kind of toxic effects. Lastly, ARIA, so amyloid therapy-related imaging abnormality is becoming increasing. There's an increased awareness of that, largely due to what we have seen as a side effects for anti-amyloid immunotherapy, right? So we have recently published paper found that it's not microglia, but rather than the perivascular macrophages are critical in driving this kind of ARIA phenotypes after immunotherapy. And also, it's quite interesting from Alector Therapeutics, where they're currently testing a TREM2 agonist in AD patients, where they actually saw, it's not an immunotherapy against A-beta, but directly activating TREM2 can also result in the ARIA phenotype, and arguably even more severe than what we have seen with the immunotherapy, right? So I think this is also further argues that the TREM2 in macrophages might be also important for some of the pathologies we see in Alzheimer's disease. So together, I think this is potentially implying a complex and in a young role of TREM2 mediated microglia response in Alzheimer's disease. I think this is really probably cause caution when we think about targeting microglia in such a broad stroke type of manner, right? Like this kind of Heimer type of approach, right? So I think we probably also want to think about the timing, when do you want to start this kind of intervention, as well as can we identify therapies that only targets specific anatomical regions of the brain, for example, the gray matter, right? Because or spared of some of the response in the white matter. So with that, I want to thank you everyone that have been involved in the studies, and also want to thank your attention. And I'm happy to answer some questions.
Jenny.
I don't know if you can tell me this, but does your antibody block soluble TREM2 shedding?
It doesn't.
Guy, and then in the middle.
That was a great talk, thank you. I mean, TREM2 can have a function just as a phagocytic receptor, and it can have another function in terms of inducing the DAM profile. But I was just wondering whether the increased NFL and increased degenerating axons could result from less phagocytosis of debris, as opposed to the opposite kind of thing, that it's promoting the degeneration itself. Do you have any? Yeah,
I think you're spot on this. So I didn't have the time to go through this. So if you look at the microglia phenotype, in this longitudinal study, where I showed the data with NFL early on, right? You see this elevation, but we couldn't detect any microglia activation differences, right? So it's only till this very late stage, 2 years period, where you start to see bona fide axonal degeneration. This is where you can start to detect the DAM phenotype. So I think this really tells us there's multiple functions of TREM2, right? As you suggest, it could be a debris removal function that's independent of this kind of DAM microglia activation. So, but this is something that we're still trying to understand, particularly from a signaling perspective. So there's a big speculation that the TREM2 has two types of signaling pathway. One is this tonic signaling, right? So this is where they keep the microglia homeostasis, which is essential for this kind of phagocytic, for example, the phagocytic functions. But it's also important having a more, I guess, what you call this, a more clustering effects of TREM2 receptor that leads to a more dramatic type of signaling pathway, which is important for this DAM microglia formation.
I think Tim's got the microphone, but then we need to go to Michael in the middle.
Sure. Thank you very much for the talk. I was wondering, because obviously there's this progression from amyloid to tau, and then during disease, it starts somewhere, it moves somewhere else, and therefore they'll be potentially at different stages, right? So does that mean that in some areas of the brain, at any given moment, TREM2 will be good and some it will be bad, and therefore do you think that we need to somehow be targeting, specific regions with TREM2?
That's the complexity, right? I think the good news here is that now we have a therapy that can remove 99.9% of the amyloid, right? So you only have to deal with tau, essentially. But still, I think, there's probably a difference between white matter and gray matter. So I think how do you find a therapy that only affects, like say, for example, gray matter microglia? This is something, I don't know if you can do that. Maybe you can.
So I think we've got time for one last question.
Really nice talk. A lot of food for thought for other drug discoverers. I'm sure you're aware there's been some recent controversy in the role of TREM2 and tau, kind of clearance, etc. There was a recent paper equally suggesting that actually TREM2 activation activity is required for phagocytosis and mitigation of tau pathology, not on the same timescale as what you're talking about. And obviously, I just wondered what your thoughts were on that.
Yeah,
you're probably aware of that work, I'm sure.
Yeah, so I think this is also similar to, yesterday Michael's talk on this kind of nanotubing, right? So really the question here is that what's really the fate of those kind of tau aggregates once they get phagocytosed? So with this, I think my speculation is the lysosomal function likely to be important, right? So if you have a really strong lysosomal function, you likely can degrade those tau aggregates, right? So maybe this in this case is beneficial. But on the other hand, we know that the lysosomal functions is impaired during aging, and also there's genetic risk factors that impairs lysosomal functions, right? So what would you do if you can't degrade those aggregates? You spit it out, right? So I think this could potentially explains why maybe those microglia actually can, in certain cases, kind of helps with the spread of the tau, right? So I think this is really, I mean, we don't have any hard data on this, but it's just a speculation. Maybe it's worth to do some experiments on it.
Okay, great. Thank you for the great questions. Thanks, Yaming.
All right.